Hello students, and I welcome you all to live TA session for environmental biotechnology. So we are in the week 6 of this course, currently dealing with problems related to lecture week 6, in which Professor Pinakisar has covered the methods that are used in microbial ecology. Yeah, it's always this class taken by me. My name is Ashutosh Behra. Is on Saturday 3 to 5 p.m. every Saturday till the start of this course, till the end of this course. So anyone who is trying to join this link will be allowed to do so. And meanwhile, I'll start the class. So basic set of instructions for all those who are participating in this live class. This is a recorded class. The recordings for this will be available on YouTube, and the link will be provided to you by NPTEL authorities, right? And the presentation or the material that I'll be sharing with you guys here on the live class will also be provided to you for your reference. These are nothing but the questions that are designed by me and old assignment papers, which will help you solve the real assignment and quizzes and help you in your online examinations, and also to understand this course better. So this is the following format that we are going to follow as this course progresses. So what I have prepared are a set of 10 to 15 multiple choice questions in the similar way you receive in your assignments or quizzes. So these are multiple choice questions and one answers are correct. Only one answer is correct. We will go through each question briefly. I will post it for the students to read the questions. I will read it aloud, and the students have to let me know what do they think is the correct answer. What is the correct answer? You have to tell me. And even since this is a recorded class, I would also urge uh, all the people watching in the comments, watching in the YouTube, please post it in the comments and let me know what do you think is the correct answer. Then briefly, for each question we will understand the logic and concept behind this, and thereby clearing your doubts and refining your concepts on these topics. Okay. And general set of instructions for students in the live class: please turn off your mic and video chat during this presentation. Let me be the only one who speaks and presents in the presents the presentation, while you can interact with me via the comment box. The chat box, I'll be regularly monitoring it for the answers and any doubt related to this week's assignment session. So assignment session is happening on Saturday 3 to 5 again, and anyone who tries, I would encourage to join the live class and clear your doubts, interact with me, and get a first hand experience of some example questions. Okay, it's already past time, and let me share my screen for lecture week 6 of the assignment. Okay, I hope every student can see my slides and hear me clearly. Can you see the slides changing? Just let me know in the chat box. Yes, thanks, Olivia. Okay, seems like we are thin on the class population today. Not many students, but that's not going to stop me from presenting and teaching you guys. So let us start this session. Once again, I welcome you all to Environmental Biotechnology live TA sessions. This course is taught by Professor Pinaki Sar from IIT Kharagpur, and my name is Ashutosh Behra. I'm a PhD PMRF scholar at IIT Kanpur. So PMRF has joined hands with NPTEL to deliver teaching deliverables in which PMRF scholars like me are taking the TA-ship duties for NPTEL courses, so that students in the NPTEL courses can have quality can have quality tutorship and benefit from this interaction, right? So we are dealing with lecture week 6. I hope you had gone through your lectures and taught by Professor Pinaki sir. And we will be dealing with multiple choice questions again. So here is the first question for this session. Question number 1. This is a match the following question. Which of the following is correctly matched? Which of these options are correctly matched? Option A, who is here? Systems level understanding. Option B, what are they doing? Phenotypic potential. Option C, what can they do? Community function. Option D, what are they doing? Community composition. Option E, what can they do? Phenotypic potential. So there are 5 columns, and the hint is this question is based on ecological communities. What kind of questions are addressed in an ecological community? I hope everyone understood this question. Let us take some time, a minute or so, and post your answers in the comment box. And for those who will be watching this recorded video, please post your answers in the YouTube comment section, and I will be checking those as well. Okay, I think it's past 1 minute, and time to reveal what is the correct answer. Let us know what is the correct answer to this question. First, let me check the comment box if students have answered. Saida is answering option C, Olivia option E, and Charna, sorry, option D. Okay, let us see which of the following is the correct match and the correct answer to this question. Correct answer is option E. What can they do is the phenotypic potential. So option E is the correct answer. Olivia has answered correctly. Well done, Olivia. Saida and Chana, you will have more chances to answer it correctly. We have so many more questions to follow. Okay, let us understand the different groups present in an ecological community and what do these terms actually mean. So the question who is here? So there are mainly three questions that three questions that are asked here in the options. Who is here? What they are doing? What can they do? These are the three questions. Let us evaluate each question one by one. Who is here? The question, the answer to this question tells us the community composition. What kind of species and composition is present in a community? If we look at a community ecology which is much larger in a scale size, then we have all these organisms performing constituting the community. And who is here? Like the secondary predators, primary predators, herbivores, plant, decay organisms, these form a community composition, and the who is here question answers these questions. Example, owl is here, rabbit is here, plant is here. And if you take a microbial community, as we see it in the course, sequencing the 16S rRNA metagenomics will help us identify the species. To answer this question, who is here? we get the answer as community composition. What can they do? What can they do? So these two questions can be a little confusing. What can they do and what are they doing? What can they do? This represents potential. Can they do, it means what can they be possibly achieving? This is the potential of the community. So this refers to phenotypic potential, the genetic capacity of the microbes. What are they capable of doing if conditions allow? So not in the present conditions, but what if optimal conditions are provided? What is the maximum extent of phenotypic potential of this community? So what can they do is phenotypic potential. For example, metagenomics to see what metabolic pathways are encoded in the DNA. And what are they doing? What are they doing as in the present tense, right now and right here in the present conditions, what is the community doing? So this is the community function. What genes are actively being expressed, what metabolites are being produced in the sample that you have isolated. For example, you can use metatranscriptomics and proteomics to see what is the current status of the community function of the sample and what can be the extrapolated, that is the phenotypic potential. So what is phenotypic potential? Phenotypic potential refers to capacity of a community's constituent organisms to modify their phenotypes in response to diverse and changing environmental conditions, allowing them to adapt and survive in their local ecology and perform variety of functions. So here is a schematic showing metagenomic analysis of answering these two questions. The same metagenomic analysis can give us the composition of the community composition. Who is there? All these animals are there, or all these microbes are there, and what are they doing? It can also give us the community function of the sample. So I hope it is clear to the students that what is community composition, which question is answered by community composition, what can they do is phenotypic potential, and what are they doing is community function. So that is why what can they do phenotypic potential, option E is the correct match. What are they doing cannot be community composition. What are they doing is community function. Community composition is who is here. Is it clear to the students? Just let me know in the chat box, or if you want to repeat, or if you want me to repeat, then I can. Very good. Thanks, Saida and Olivia, and Charna as well. Okay, moving on to the second question. Question number 2. Which of the following researcher discovery pair is incorrect? Again, a match the following type, but this is a difficult one. Which of the following researcher discovery pair is incorrect? Option A, Carl Wu's 16S rRNA based phylogenetic analysis. Option B, Norman Pace rRNA based phylogenetic analysis. Option C, Robert Koch pure culture technique. Option D, Sergei N. Vinogradsky culture independent analysis. And option E, Staley and Konopka great plate count anomaly. Okay, here are the options before you. Think about it. Which scientist is matched incorrectly to their discovery? Only one of these is correct. Let us take some time and answer these questions. And those who will be watching this YouTube video, also try to think about it and post your answers in the comment box. Okay, I think it's time for question number two to reveal what is the correct answer to this question. Let me check the chat box. Saida, Charna, and Olivia have all answered option D. Okay, and if you say so, let us look at which is the correct answer, which is the correct researcher discovery pair. Right? Option D is the correct answer. All of you guys are correct. Congratulations, Saida, Charna, and Olivia. Option D is the correct answer.
Stop taking the name. Stop it.
Hello, Olivia is saying something, I guess. Yeah, Olivia, if you can write, if you have any questions, you can post it in the chat box. Okay, okay. So Sergey and Vinogradsky and culture independent analysis is the incorrect pair. Vinogradsky is well known for his discovery on Vinogradsky column or isolating bacteria. Okay, let us pick up a few scientists and their innovations and understand it correctly. So Carl Woos and 16S rRNA gene sequencing analysis. This is Carl Woos on my right. He pioneered the use of 16S ribosomal RNA to establish three domain system of life, which is archaea, bacteria, and eukarya. So mainly for the microbes, 16S rRNA is the gold standard for any metagenomic analysis as well. So if you look at what is 16S rRNA, 16S rRNA is a part of the prokaryotic ribosome. The prokaryotic ribosome is a 70S. S stands for sedimentation constant. 70S subunit ribosome. It has two subunits, large subunit and the small subunit. In the large subunit is composed, so each ribosomal subunit is composed of rRNA, which is ribosomal RNA, and ribonucleoproteins and ribosomal proteins as well. So in the large subunit, the 50S large subunit, 5S rRNA and 23S rRNA is abundant, while in the small subunit 30S1, 16S rRNA is abundant. And then these two components are assembled to form a complete ribosome. So why 16S rRNA? Because there are various conserved regions and variable regions which tells us about the ancestry and evolutionary divergence of archaea, bacteria, and eukarya. So Carl Woers in 1977, his work revealed that prokaryotes were distinct lineages with archaea being more closely related to eukarya than to bacteria. So this was his discovery. Eukarya and archaea were together and bacteria was far apart. And the 16S rRNA gene's universal presence, slow mutation rate, and conserved function made it an excellent molecular clock for evolutionary studies, allowing Woos and his colleagues to chart the phylogenetic relationship among all organisms. So, this is what I was talking about. So, the universality of 16S rRNA and slow mutation rate made it a conserved sequence between all the prokaryotes, which can tell us about the evolutionary origin because the region is conserved between species. And this allowed him to dissect eukarya, archaea, and bacteria. Right? And 16S rRNA sequencing is now routinely followed in many metagenomic analysis. Now, looking at another option, which was Norman Pace. What did Norman Pace do? Norman Pace was again using rRNA for phylogenetic analysis, particularly for studying unculturable microbial communities. So, this was Norman Pace's contribution. Many microbes are unculturable, which cannot be studied in the laboratory or grown. His lab developed techniques and published influential papers on rRNA gene cloning and sequencing to build a phylogenetic map of life, especially bacteria and archaea's domains, and identify environmental microbes. Environmental microbes in the sense halophiles, extremophiles, acidophiles, thermoacidophiles, right? So his lab had tried to use rRNA based sequencing and cloning and to work on unculturable microbial communities. His work established the rRNA based approach as a powerful method in microbiology and contributed to the emerging field of metagenomics. So, Norman Pace's rRNA phylogenetic analysis helped us to map genomes after the metagenomic sequencing. Then coming on to Sergio Vinogradsky. Sergio Vinogradsky is famously known for Vinogradsky column, which is named after him. Is credited with discovering biological process of nitrification, the first known form of chemoautotrophy, and for developing Vinogradsky column for culturing microorganisms. So this is also the scientist who discovered the nitrification process as we know now, the great nitrogen biogeochemical cycle, and different bacteria which play a role in nitrification. And what is the Vinogradsky column? Why is it so famous? This is a simple self-sustaining ecosystem created in a clear container filled with mud, water and nutrients. So using naturally and easily available samples, this column can be created, in which, due to oxygen and light gradients present in the column, different microbes dominate at different depths, such that top zone is the aerobic zone where most oxygen and air is available, the top zone of the column. Cyanobacteria, algae do photosynthesis, and there are aerobic bacteria. The middle middle of the column is microaerophiles or anaerobic transition or facultative anaerobes. Purple sulfur bacteria using light. The light can penetrate, but the oxygen is not here. And H2S as a donor, since oxygen is not there. Purple non-sulfur bacteria. And the bottom zone are anaerobic zones, no oxygen and high sulfur. Green sulfur bacteria, sulfate reducing bacteria, fermenters and methanogens. So what did Vinogradsky did? Vinogradsky took mud, water and nutrients in a jar and covered it with a plastic wrap and kept it for months and allowed the microbes to grow. So microbes separated on the basis of oxygen and light gradients, showing us this distribution, the top zone, middle and the bottom zone. So Vinogradsky is not known for culture independent microorganism growth, rather than for this separation technique in the Vinogradsky column, and also for nitrification, discovering the nitrification of the nitrifying bacteria. Okay, is it clear to the students? Just let me know. Since you have all answered correctly, I think you might have known this answer. Very good. Good. Thank you for the response. Moving on to the third question. Third question of this session. Greater than 99% of all microorganisms cannot be cultured. Which of the following is not a probable explanation for this? So many microbes cannot be cultured. What could be a probable explanation? What is not a probable explanation? Okay? It means one of these is the wrong explanation and all others fit into the explanation. Option A, these microorganisms are not viable. Option B, these microorganisms are dormant. Option C, these microorganisms are mutants of culturable strains. Option D, we do not have laboratory growth medium or conditions suitable for them. Option E, these microorganisms cannot survive in pure culture without microbial interaction. Now, which of these is not a probable explanation why greater than 99% of all microbes cannot be cultured? Understood the question? Okay. Let us take some time and collect answers in the chat box. I will be visiting the answers in a while. Okay. Okay, I think it's enough time for this question. Let us see what is the correct answer to this question, and let us see what people have answered in the comment box. Charna has answered option A. Okay. Olivia is confused between A and B. Saida is answering option C. Okay. So I hope you have guys have all understood this question. The question is asking which of the following is not a probable explanation for this. For what? For greater than 99% of microbes cannot be cultured. Okay. So correct answer to this question is option C. Option C is not a probable explanation for this. Microorganisms are mutants of culturable strains, so they cannot be cultured. So this is not the reason why greater than 99% of all microbes cannot be cultured. So I think Saida is correct this time. Very good. And Chana and Olivia, you can, I'm sorry, but in the next question you will get a chance to be correct. Okay. Let us understand each of these options and clear our doubts about this. Now, what are the key reasons for culturing a microbe in a laboratory setting? What are the key reasons? Key reasons include the absence of specific nutrients or growth factors. Why is not the microbe being cultured in the lab? Because it does not have specific nutrients and growth factor for its survival. So many microbes depend on specific nutrients and growth factor. They also depend on symbiotic relationship with other microbes and adaptation to extreme or slow growth environments and the inability to replicate necessary micro environments. Right? So number 1, microbes can have specific growth or nutrient factor requirement. That is why they cannot be grown, which is corresponding to option D. Right? Microbes often have symbiotic relationships or interactions with other microbes in the community. Thereby, a single microbe cannot be isolated in the lab. Many microbes. So, these microbes cannot survive in pure culture without microbial interaction, covering option E. They adapt very slow to slow growth environments, extreme and inability to replicate necessary micro environments. So we cannot exactly replicate the micro environment of the microbe. That is why the microbe is not able to go. With precise nutrient gradients, oxygen levels and osmotic conditions which exist in the wild. And also there are a ratio of viable and dead cells. Right? And in the viable category, cells can be also in dormancy, which makes them unculturable. Now, what is dormancy? The cell is alive but is not active, it is metabolically inactive, and if such a cell or a microbe is picked up, it cannot be grown in a culture media. While the culturable phases include lag phase, exponential, and stationary phase, cells in these growth phases can be cultured, but cells in dormancy are unculturable. So, this covers microorganisms that are dormant and microorganisms that are not viable. Of course, dead cells or dead microbes cannot be cultured. So, what are these possible explanations? Going through it again. Option A, microorganisms are not viable. Sometimes true, because dead or non-viable cells will not grow. But this does not explain the majority of 99% of microorganisms. Option B, these microbes are dormant. This is also true because many microbes exist in dormant, viable but non-culturable state. And option D, we do not have laboratory growth medium conditions. Yes, because many need specific nutrients or conditions. And option E, microorganisms cannot survive in pure culture because many rely on syntrophic and symbiotic partners. So the odd one out is option C. This is not a general explanation because vast uncultured diversity is not just mutants of known strains, they are entirely different taxa with unique physiological requirements. Yes, this is another point I wanted to focus on. Bacteria in a community are just not the mutant version of each other, while there are many taxa and many phylogenetically related many species of bacteria present in a sample or a community. That is why with unique physiological requirements. So that is why this is not a general explanation why many organisms are not culturable microbes, because many mutants are also culturable on agar plate or on lab cultured methods, but this is not a suitable explanation for the majority of microbes being uncultured. While these are the possible explanations, microbes can be live or dead, microbes can be dormant, which are alive but in non culturable state. They depend on molecular interaction with other partners, and also laboratory settings cannot replicate the exact environment and growth media in the wild in which the microbe grows. That is why option C is the correct answer, which is not the valid general explanation for this question. I hope it is clear to the students. Please tell me in the chat box, and we are good to go. Yes, sir. Okay. Thank you. Thank you, Olivia and Saida. Moving on to the next question, question number 4. Question number 4 is about enrichment culture. Enrichment culture is used to dash. Option A, enhance total microbial community. Option B, enhance specific sections of microbial community. Option C, selectively allow growth of specific microorganisms. Option D, selectively allow bacteria to grow. Option E, visually distinguish groups of microorganisms. What is the purpose of enrichment culture? This is the question in a nutshell. Let us take some time and arrive at some answers in the chat box and in the YouTube comments as well. Tell me what you think, students, is the correct answer, and I will revisit this question in a minute. Okay, I think it is time for me to reveal what is the correct answer. Let me check the comment box for any answers by the students. Saida, Shanna, and Olivia have answered option C. Okay, let us see what is the correct answer to this question. Option B is the correct answer. Enhance specific sections of microbial community. But reading together, option C is also meaning the same thing, to selectively allow growth of specific microorganisms. So I would change this to option B and C. Rare occasion in which two options are looking very similar. Actually, the definition of enrichment culture encompasses these two. So you guys are correct. Option C is also the correct answer. Let us understand what is enrichment culture, and I will change this. So enrichment culture is used mostly to isolate an organism from natural sources. Here is an example schematic: medium containing select nutrient sources chosen because few bacteria other than the organism of interest can use them. Sample that contains a wide variety of organisms including the organism of interest is added to the medium. So from a mixture of organisms you want to isolate the organism of your interest. Now organism of interest can multiply whereas most others cannot, because the medium is suitable for the organism of our interest, and thus we are enriching the organism or microbe in this process, and the enriched sample is plated into appropriate agar medium. A pure culture is obtained by selecting a single colony of the organism of interest. So this is how an enrichment culture is done, selectively promoting the growth of one microbe over the other and enhancing specific sections of microbial community. Enrichment culture is a microbiological technique that increases the population of a desired microorganism from a mixed sample by manipulating growth conditions to favor its growth and inhibit competitors. So I think option C is also favoring this answer. This is achieved by providing a specific enrichment media and environmental conditions such as temperature or nutrient availability that are ideal for target organism and unfavorable for others. So this is a comparison between various practices such as enrichment culture, selective media, and differential media. What is the purpose of enrichment culture? To increase or target microbes in abundance, selective media to inhibit unwanted microbes, and differential media to visually differentiate different microbial communities, microbial population in a community. The method, environmental medium manipulation, is the enrichment culture method. It provides specific media. While in selective media we have antibiotics or chemical inhibitors, in differential media we have pH indicators which can indicate different microbes based on their acidity, their temperature, or pH. For example, nitrogen free medium for nitrogen fixers to promote enrichment culture of nitrogen fixers, mannitol salts agar for selective media like staphylococcus, McConkey blood agar to visualize differential media. So I think correct answer is option C. Now analyzing all this, so I will change this. Option B is also correct, but mostly enrichment culture is used to selectively allow growth of a specific microorganism. So option C is the correct answer. Thank you for answering this. All of you guys have answered correctly. I will save this so that this corrected presentation can reach as a material to you guys. Right, so option C is the correct answer to enrichment culture question number 4. I hope it is clear to the guys, to the students. In enrichment culture, we are promoting a certain microorganism favoring over the others. We add selective media, we add inhibitors, and we add growth factors, environmental conditions to match or promote the organism of our interest or microbe. I hope it is clear to the students. Just tell me in the chat box and we can move ahead. Very good. Thank you, Charna, Saida, and Olivia. Looking at the next question, question number 5. This is a problem statement. You have to design the problem, solve the problem yourself. A medium devoid of organic nitrogen was inoculated with soil. Let me use my pointer. Yeah, read the question very carefully. This is a long question. A medium devoid of organic nitrogen was inoculated with soil and amended with ammonia. Understood? Medium devoid of organic nitrogen, inoculated with soil, and ammonia was provided. After an adequate incubation period, the culture was subcultured to microbial media without ammonia. With and without ammonia. So there are now two subcultures. One subculture is in a microbial media with ammonia and the other one without ammonia. Negligible growth was seen on media without ammonia, though growth appeared on medium containing ammonia. This could be due to dashed. Understood the question? It is also posted in your screens. Please read it carefully. Analyze the conditions. How was the pure culture done? How was the subculture done? And what is the result and what is the question? Negligible growth after the subculture was seen on the medium without ammonia, though growth appeared on medium containing ammonia. Now, what could be the possible reasons? Option A, lack of selective pressure for nitrogen fixation. Option B, lack of selective pressure for ammonia utilization. Option C, catabolite repression of ammonia synthesis. Option D, toxicity of ammonia. Option E, enrichment of ammonifiers. Understood the question? Let us take some time, some more time, because you might need some time to analyze these options and the question especially. So take some time and write your answers in the chat box and in the comment box. I will follow them shortly and come back to this question to tell you what is the correct answer. Okay, I think it is time for me to reveal what is the correct answer to this question. Let me let me see what is written in the chat box by students. Saida is saying option E, Olivia A or E, and Sharna says option A. Okay, so after understanding this question, we can deduce it. What is the correct answer to question number 5? Answer is option A, lack of selective pressure for nitrogen fixation. This is the reason why we see this phenomena, that negligible growth on medium without ammonia, but growth appeared in selecting with ammonia. Okay, so what actually happened in this procedure or technique, we will understand. Okay, so this was the situation. I have pictured it down from the question. First, the microbe communities were cultured in media which was devoid of nitrogen but amended with ammonia or present in ammonia. So this enriched for ammonifying microbe. So only those microbes which could utilize ammonia, they grew in this culture. And since this was devoid of nitrogen, no nitrogen fixers, there was no pressure for, there was no lack of selection pressure for nitrogen, right? So here the bacteria only which could positively utilize ammonia, they grew. And then in the subculture there are two conditions. You grow it in media with ammonia, then there is good growth, but there is a media without ammonia, then there is no growth, right? And why is the answer A the correct option? The original enrichment culture contained ammonia, so organisms that use available ammonia were favored. That removes the selective pressure for microbes that must fix nitrogen. Those organisms would not be enriched, right? So nitrogen is not the limiting factor or the selection pressure, while presence of ammonia is the selection pressure. So that removes the selective pressure for microbes that must fix nitrogen for survival. When you subsequently transfer to medium without ammonia, the culture shows negligible growth because enriched population lacks organism that can fix atmospheric nitrogen. This is true because if such organisms would have been selected, then they could have even done well without ammonia if they were nitrogen fixers. Other choices don't fit because ammonia's presence actually selects for ammonia users. So B is also wrong. And ammonia wasn't toxic because growth appeared in ammonia plates. That is also true. So option D is also wrong. And there was no organic nitrogen present to enrich ammonifiers. So option E is also wrong. So you need organic nitrogen to be present to enrich ammonifiers. Choice C is a confused phrasing. Ammonia represses nitrogenase expression, but the simplest and most correct explanation is A. So lack of selective pressure for nitrogen fixation removed the N2 fixers, and therefore in the subculture, in the absence of ammonia, there was little growth. If there were nitrogen fixers, they could have fixed atmospheric nitrogen rather than depending on ammonia. I hope it is clear to the students why option A is the correct answer. There is a lack of selective pressure for nitrogen fixation, which led to low growth in a medium without ammonia. Okay, very nice. Moving on to the sixth question. Most probable number technique is used for dash. Most probable number technique. Option A, enrichment of specific microorganisms. Option B, dilution of specific microorganisms. Option C, enhancing growth of specific microorganisms. Option D, estimating population size of microorganisms. Option E, estimating number of species of microorganisms. Most probable number. Recall this technique and tell me what you think is the correct answer. Okay, I think it's time for discussing this question. Question number 6. Most probable number technique is used for dashed. Let us see if students have answered. Saida, Charna, and Olivia have answered option D. Okay, and the correct answer to this question is option D. You all guys are correct. This is the correct answer. Most probable number is used for estimating population size of microorganisms in a culture. Right? Now, what is this technique? This is a statistical method to estimate concentration of viable microorganisms in a sample, particularly when the concentrations are very low. And it involves inoculating multiple tubes with serial dilutions of the sample into a growth medium and observing the presence or absence of growth. So we serially dilute the samples into multiple tubes to find out a concentration in which there are minimal growth of microorganisms. The pattern of positive and negative results across different dilutions is then compared to a statistical most probable number table to determine most probable number of organisms in the original sample. So here is a step-by-step procedure for performing this technique. The original sample is serially diluted into multiple small volumes. 1 is to 10 times dilution, 1 is to 100. So first you take 1 ml of the culture and then 10 ml of media, 9 ml of media, 1 is to 10. Too numerous to count. This plate, when plated, it will have lot of microorganisms. But when you do another serial dilution, 1 ml of this in 9 ml of new media, it will be 100 times diluted. So we go on doing serial dilutions until we can count the number of colonies in our fingertips and then trace back to the most probable number. Each dilution is inoculated into multiple tubes containing a suitable growth medium like LB broth. The tubes are incubated to allow for microbial growth, and the presence or absence of growth in each tube is observed. The pattern of positive tubes in each dilution series is recorded, and this pattern of positive tubes is then matched to statistical MPN table to find the most probable number of microorganisms. So this is the technique which is primarily used to identify the count or the number of microorganisms even in low concentration cultures. Since you guys have answered this correctly, I think it is clear to everyone. Please let me know in the comments and we can quickly move to the next question. Okay, thank you for the approval. Moving on to the seventh question. Which of the following statements comparing direct and enrichment based isolation of bacteria is incorrect? Which of the following statements comparing direct and enrichment based isolation of bacteria is incorrect? Option A, direct culture increases chances of getting the desired subgroup of microorganisms. Option B, direct culturing reduces biases introduced by enrichment technique. Option C, lab weeds can outgrow desired groups more easily during direct culture. Option D, different enrichment media would be required for every distinct target subgroup. Option E, isolating pure culture from enrichment culture will lead to loss of at least some microbial species from the culture. You have to tell me which of the following statements is incorrect based on a comparison between direct and enrichment based isolation of bacteria. Okay, I think it is time to look back at this question. Which of the following statements comparing direct and enrichment based isolation of bacteria is incorrect? Let me check the comment box for answers. Charna says option A, Olivia and Saida option A. Okay, let us see what is the correct answer to this question. Direct culture increases chances of getting the desired subgroup of microorganisms. This is the incorrect option, that is why this is the incorrect statement. So it's the correct answer option A. Charna, Olivia and Saida all have answered the correct one. Well done. Let us understand the difference between direct and an enrichment based isolation of bacteria. So on our approach of direct and enrichment based isolation, for direct isolation, plating directly onto the medium. For enrichment based isolation, modifying the conditions to favor only a certain type of microbes to grow. What is the bias? There is no bias, low bias, but misses the rare ones in the direct isolation. While in enrichment based isolation, we have high bias for selecting specific kind of microbes. What are the microbes recovered? They are abundant microbes recovered and fast growers in direct isolation since there is no inhibition. While in enrichment based isolation, rare and specialized functionally important microbes are enriched. For example, dilution plating of soil or pore plating method. While enrichment based isolation, nitrogen free medium for nitrogen fixers and Vinogradsky column. Both of these examples we have encountered in the previous questions. Usefulness: direct isolation gives us a community overview, what kind of microbes are present in the whole community, while enrichment based isolation gives us an opportunity to study specific metabolic groups present in the community. Direct isolation is good for abundant and easy to grow microbes, whereas enrichment isolation is good for specific rare microbes with unique metabolisms. So on my left is a direct culture method, pour plate method, pipetting the bacterial sample onto the petri dish, pour liquid nutrient agar, swirling to mix it, and colonies grow on agar surface and subsurface. So this is the pour plate method, direct plating, and while this can be a serial dilution method for enriching bacteria in saline medium to select for bacteria that are able to utilize saline for their growth. I think it is clear to the students. You have all answered correctly. For a direct culture does not increase the chance of getting a desired subgroup. It is rather the enrichment based isolation in which we are enriching for a specialized group. Okay, thanks for the confirmation, Saida. Let's look at the 8th question of this session. Question number 8. When setting up an enrichment culture for microorganisms capable of biodegrading phenol, which one of the following steps is the most important? Understand the question carefully. You are trying to enrich for microbes which can degrade phenol. Which step is the most important? Option A, eliminating all carbon sources except for phenol from the medium. Option B, eliminating all nitrogen sources except nitrogen gas from the medium. Option C, determining the optimal pH for phenol degradation. Option D, determining optimal temperature for phenol degradation. Option E, determining ideal growth conditions to promote syntrophic associations in the culture. Now, which of these is the most important step that you will take to ensure that you are selecting or enriching for microbes that can biodegrade phenol? Let us take a minute or so and collect some answers in the chat box and in the comment box. I will visit this question later to reveal what is the correct answer. Okay, I think it's enough time for this question. Let us check the chat box for some answers. I can see there are already some answers. Option A, unanimously by Olivia, Saida, and Chana. When setting up an enrichment culture for phenol biodegradation, which of the step is important? Option A is the correct answer. Eliminating all carbon sources except for phenol. That is how you can select for bacteria which can degrade phenol, which can survive in the presence of phenol. Right? So option A is the correct answer. Congratulations. Well done, everyone. So let us look at this schematic and what is the key principle for selecting an enrichment media. To isolate microbes with a specific metabolic ability, you provide that substrate as the only usable source of energy or carbon. This creates selective pressure so that only microbes that can use it will thrive. So this is actually from a research article in which they have tried to select for phenol degrading bacteria. Lake bed sediments 500 milligram of phenol in this culture, and they are enriching this culture with high amounts of phenol so that only bacteria with multiple passages, only those bacteria will survive which can utilize this high amount of phenol as their only carbon source, and then using batch experiment in different conditions to find out which are the groups of bacteria represented in these communities that have survived such high concentration of phenol. So to isolate microbes, we only provide that substrate which can be used as a carbon source to create a selective pressure that can enrich microbe which can degrade the phenol. So thereby checking all options, Eliminating all carbon source except phenol, this is the correct option. Eliminating all nitrogen source except nitrogen, that would enrich for nitrogen fixers, not phenol degraders. This which we saw in question number 5, in which we had a problem related to nitrogen and ammonia fixers. Option C, determining the optimal pH for phenol degradation, important later for optimization but not for selecting the bacteria. You can, once you select the bacteria, you can tweak the pH and the temperature and the chemical nature or the enzymatic activity of the bacteria for optimal phenol degradation. Determining optimal temperature, same as option C. Determining ideal growth conditions, this is relevant for some anaerobic degraders but not the first most important step. So the question was about the most important step, which is replacing all other, exhausting all other carbon sources and using phenol as the only carbon source. I hope it is clear to the students. Tell me in the chat box and we will move ahead to the next question. Very good. Okay. Moving on to the next question. Question number 9. This is the second last question. I have prepared 10 questions for you guys. So, two more questions to go. Question number 9 and question number 10. Okay. Question number 9. General staining methods do not provide information about microbial metabolic traits. Which one of the following modifications would help us to gather this information? Any staining method which can help us to identify metabolic traits of the microbial community. Option A, fluorescent dyes that can distinguish between intact, which are viable, and ruptured cells. Option B, fish using DNA probes complementary to genes of interest. Option C, cardfish using DNA probes complementary to mRNA of interest. Option D, cardfish using DNA probes complementary to the rRNA of interest. And option E, radio labeling of genes of interest. Understood the question? We have to find out the metabolic footprint of the ecological community or microbial community. Which one of these options is the most suitable method? Let us revisit this question in a minute or so to reveal what is the correct answer. I think it's enough time for this question. Question number 9. Let us find out the correct answer. First, checking the comment box for student answers. Charna option C, Olivia option C, and Saida is also saying option C. And I'm happy to announce that option C is the correct answer. Again, all of you have answered correctly. Well done. Card fish using DNA probes complementary to mRNA of interest. And let us understand why is it so. Why is it this answer the correct answer? So even before moving on to the explanation, I want you to notice the difference between DNA, the mRNA, and if a cell is alive or not, right? Now the question asks, we want to understand what are the metabolic traits. Now metabolic traits are a function of the proteins or pathways, metabolic pathways that are going on in a community. And this is a signature of the different enzymes and proteins that is expressed in the community. Now which molecule is the proxy, or which is the proxy for all the proteins expressed in a community? It is the mRNA. This is the messenger RNA that is translated into the protein coding, into the protein structures. Thereby, probing the mRNA will tell us which are the genes that are getting expressed, or which are the enzymes that are actually functioning, and which will tell us the microbial metabolic traits. That is the question of our interest. Okay. Now, what is this procedure, cardfish? Cardfish works through this technique. First, you take the sediment sample or the microbial sample, fix it in paraformaldehyde or PFA, cross-linking all its mRNA and proteins in same place, then permeabilize the cells with lysozyme or an enzyme to rupture and permeabilize the cell, not rupture. After cell lysis, cell pores are bigger and target rRNA or mRNA is accessible for the probe binding. Then you inactivate endogenous peroxidase in the cell by treating it for quenching the superoxide radicals and the peroxide radicals, hybridize the specific 16S rRNA or mRNA probes, oligonucleotide probes, to these ruptured cells at 35 degree, and afterwards removal of non-binding probe with wash the excess probe, accumulation of tyramide radical with fluorescein attached on HRP after catalysis. So addition of the substrate in which horseradish peroxidase, which is attached in the probe here, will then convert this substrate into a color substance, and visualization by fluorescence microscopy. So an oligonucleotide probe with an HRP enzyme attached targets a specific mRNA sequence within a microbial cell. Signal amplification: in the presence of hydrogen peroxide, HRP enzyme converts tyramide into a reactive radical form. These tyramide radicals then bind non-specifically to nearby aromatic compounds such as tyrosine and tryptophan within the cell. A large number of these tyramide molecules get deposited near the HRP, creating a highly amplified and sensitive fluorescent signal that makes the targeted microorganism visible under a microscope. Sorry. Okay, so this is a technique of cardfish, or the procedure of cardfish. Now, what is the problem with the general stain? Standard stains like gram stain or viability dyes only can show cell structure but not metabolic activity. To study metabolic traits, we need to examine gene expression or mRNA or the functional RNA or protein activity. This is what I was trying to say. So then evaluating all other options, fluorescent dyes distinguishing intact and ruptured cells. So again, live and dead cells is not the correct way. Fish with DNA probes complementary to genes of interest. It can only target the genes but not the expression of these genes. DNA presence only indicates potential, not the actual activity. Cardfish with the mRNA is the correct answer, while where card is catalyzed reporter deposition, the method in which HRP and tyramide are detected by depositing lot of tyramide near the HRP tagged probes, increasing sensitivity. Cardfish with DNA probes to rRNA will only tell us about the different type of microorganisms present, but not the actual function. rRNA abundance correlates with general activity or growth rate, but not specific metabolic traits. And radio labeling of gene of interest, this is also not, because such methods are used for in situ detection of metabolism, but will not tell us the actual activity or gene of expression uh, dynamics, which can only be accessed by cardfish against mRNA of the microbial community. I hope it is clear to the students, since you guys have all answered correctly. Cool. Thank you. Moving on to the last question of this session, question number 10. Which of the dyes or combination of dyes can be used to differentiate live and dead cells? A related question. Which of the dyes or combination of dyes can be used to differentiate between live and dead cells? And the options are option A, propydium iodide, option B, AO, option C, DAPI and AO, option D, cytonine, propydium iodide, option E, DAPI and cytonine. Tell me the correct answer in the chat box. Okay, I think this question does not require more time. This is just a knowledge based question. Which of these dyes and combination of dyes can be used to differentiate live and dead cells? Olivia, Charna, and Saida are saying option D. And option D is the correct answer. Cytonine propydium iodide is the dye combination used to differentiate between live and dead cells. So this is a dual staining method where cytonine can penetrate all the cells and stains DNA in green color, while propydium iodide penetrates only the cells which have damaged cell membranes, or in short dead cells, which can stain the DNA in red color and quenches the cytonine fluorescence. So then in a mixture of cells, green glowing cells are the live cells, while red glowing cells where green fluorescence is quenched is the propydium iodide entered cell and or the dead cells. So live cells will be green and dead cells will be red. So propydium iodide only stains dead cells. Option A is incorrect. Without a counter stain, acridine orange only stains nucleic acid but does not differentiate live versus dead. So you need to pair the acridine orange with the propydium iodide to do a dual staining to identify live versus dead. DAPI plus acridine orange, both will stain DNA but not a viability test. Cytonine PI is a classic light dead backlight assay, and DAPI cytonine again both stain DNA, not useful for viability distinction. So what happens with these two dyes, cytonine and propydium iodide? Core principle relies on the differential permeability of two fluorescent stains to bacterial cell membranes. The cytonine is a green stain. This stain can readily penetrate all bacterial cell membrane, mentioned here in green. Regardless of their integrity, when used alone, it stains all bacterial cells, giving a measure of total cell population. So cytonine, it can go cross the cell membrane of bacteria and stain the bacteria green. While propydium iodide is a red stain, is only able to enter bacteria when the cytoplasmic membrane is damaged or compromised, which is a characteristic of a dead or dying cells. So this is why this dye pair is used for a live dead assay to analyze how many microbes in a community or a sample are alive and how many are dead. I hope it is clear to the students. Very good. So with this we have come to the end of our live TA session. So it was fun to solve these questions with you guys, and you guys were actively participating. That is always a good thing and rewarding to a teacher as well. So if you have any suggestions for improvement of this class to include more questions or the style of presentation, you can let me know in the chat box. Or if you have any doubt you want me to solve it, please tell me right now, or you can tell me in the next class. Please go through your lectures. I hope you liked the class. And again, this will be a recorded lecture and available to all of you guys via YouTube. And see you in the next class with lecture week 7. And I'll be ending this call. Thank you for joining. And I would encourage all the students and your friends who have joined, or if they have taken this course, please ask them to join the live classes and take the maximum benefit out of it. Okay. Thank you. I will end this call. Thank you for joining. See you soon next Saturday with lecture week 7.